Hi, I'm Rachel Kurlander, and my first case connections problem I will be talking about is problem number 2, which first asks about how glucose uptake is regulated in normal liver, brain, skeletal muscle, and adipose tissue. Then follows up with how does normal glucose uptake change when there is a tumor located in the brain, and how would it benefit the tumor? And finally wants to know what changes happen in the cancer cells to alter the uptake of glucose. So first, in a normal healthy liver, glucose uptake is regulated with glucose transporter GLUT2 and an insulin-independent mechanism to move glucose down a concentrating green across the cell's membrane. Skeletal muscle and adipose tissues use GLUT4 in an insulin-dependent manner, while the brain uses GLUT1 and GLUT3 in an insulin-independent manner. Cancer cells cause an increase in glucose dependency due to needing more energy in order to develop masses or tumors. Cancer cells will purposefully use glycolysis to generate ATP even in an aerobic environment. Certain cancer-causing genes can even cause hyperactivity in glutes and an increase in the fermentation from glucose to lactose. This is called the Warburg effect, which uses the increased amount of glucose present within the cell, which uses an increased amount of glucose present to help with the cell proliferation into tumors. The Warburg effect also indicates that with an increased rate of glycolysis, there's an increased rate of lactic acid fermentation in the lactic acid cycle as opposed to pyruvate dehydrogenase, which is turned off in cancer cells. During this effect, glycolysis and the citric acid cycle are uncoupled as well. Once there is an excess of glucose, the cancer cells send it to the pentose phosphate pathway to create more NADPH and ribulose 5-phosphate to stimulate more cancer cell growth. As NADPH will help with the synthesis of fatty acids, and ribulose 5-phosphate will help in the synthesis of nucleotides, thus providing cancer cells with the nutrients needed to continue into new cell creation. By uncoupling glycolysis in the citric acid cycle, cancer cells were able to alter cell signaling pathways to fit their need for fast energy production as well as cell creation. There's also a phenomenon called the Crabtree effect, where there is a lack of oxygen present in glycolysis, allowing for rapid growth of cancer cells. Cancer cells can also cause a shortage of ATP when using glycolysis in excess. This happens because hexokinases will start to convert chemicals similar to glucose, but they are unable to be used in the rest of the cycle, which leads to a stop in glycolysis. These changes in cell metabolism have also been linked to the activation of certain cancer-causing genes, or oncogenes, and the repression of tumor-suppressing genes, thus allowing for optimal environment for cancer cells to grow and spread. The next case connection I chose was number 4. Which was describing a patient, a 2-month-old female, who was experiencing jittery periods after feeding on breast milk. The patient also has an enlarged liver, indicated by percussion in the abdomen, as well as trouble maintaining blood glucose in the fasting state. When given epinephrine, there is an increase in blood glucose, indicating a problem with glycogen breakdown. The problem then gives 4 possible diagnoses, which include hereditary fructose intolerance, galactosemia, glycogen storage disease, and glucose 6-phosphate dehydrogenase deficiency. Some common symptoms of hereditary fructose intolerance include vomiting, sleeping for long periods of time or very frequently, or failure to thrive. These diseases would also require both parents to have a recessive copy of the gene to have been passed down to the baby. Test results would also confirm a high level of uric acid and a low level of blood sugar. While the baby is sleeping for more than 2 hours between feedings, she is not vomiting, nor is there any mention of her losing excessive weight or not eating at all, which would likely rule out hereditary fructose intolerance. There is also no mention of uric acid levels in any of the tests, and after feeding her, her blood sugar levels were normal. She only had problems maintaining them in the fasting state. The next possible choice was galactosemia, which has symptoms including vomiting, jaundice, and poor activity levels. Patients who have this disease tend to suffer damage to the liver, brain, and eyes as well, This disease is also recessive, so both parents would have had to have had a copy of this gene. People with this disease are unable to tolerate any sort of milk at all, and this disease can also be caused by infections of E. coli in the first few days of life, either through a dirty bowel or through contact from another person that may have had it on their hands. And this can be found with a bacterial culture. Signs of this disease also include ketones in the urine or found in urine samples and normal to low blood sugar level directly after feeding. The baby in the case study has not shown symptoms of vomiting, jaundice, poor activity levels, or problems with her brain or eyes, and there was no mention of a urinalysis, which indicated we could rule out galactosemia out as well. The next options are glycogen storage disease and glucose-6-phosphate dehydrogenase deficiency. Glucose-6-phosphate dehydrogenase deficiency symptoms include fever, fatigue, dizziness, a quick or rapid heartbeat, paleness, and shortness of breath. G6PD is also a genetic condition, meaning one or both parents would have had to have this deficiency, and since she was a female, both parents would have had to have this as a recessive form. But this was also not disclosed in the parent's case. The disease can eventually turn into anemia if not on a proper diet or treated early. The lab tests for these include complete blood counts and serum tests. There was no mention of a complete blood count or serum test ran on the baby, indicating that this was not likely a possibility. The baby also did not have a fever, shortness of breath, or paleness noted in her symptoms. And the symptoms also only show up when the patient has ingested something that could attack red blood cells, and in this case, she has only ingested breast milk. The final option is glycogen storage disease, which is most often found in infants and young children. This disease happens when glycogen is unable to be broken down in the liver and turned into glucose, which can cause an excess of glycogen in the liver. When the patient was administered epinephrine, it failed to turn glycogen into glucose, which is a key factor in diagnosing this disease, as epinephrine, when in a proper working system, should help boost the breakdown of glycogen into glucose. This disease is also hereditary, but it is recessive, so both parents would have needed this gene for the child to get this disease and show symptoms, while the parents would not have any signs of glycogen storage disease. Symptoms of this disease include hepatomegaly, an enlarged liver, hypoglycemia, low blood sugar, swollen abdomen, cramps, lactic acidosis, hyperuricemia, and muscle weakness. Most of these symptoms do align with the symptoms the baby was experiencing, leading me to choice number C, glycogen storage disease, being the baby's diagnosis, as this one was the one that seemed to have the most symptoms in line, and seeing that the test could fit the same as what was given in the information in the case study.